If any one of you has any doubt and any question, you feel free to ask us in the chat box.
Hello. So, hello everyone. Hello students, all the learners. Welcome to the live session. Am I audible?
Yes, madam, you are audible.
Yes, ma'am.
Okay, very good. So we have a bunch of students joined here. Hello Shan, Anupama, Tanishka, Praveen, and Itishri, Pratik, Astha, Samiksha. A lot of you have joined, so very good. Basically, in this live session, it's open to you if you have any doubts about the course, the progress, um, and so on. You can just excuse me. So we are actually connected on this forum, so you can type your questions in the chat box. That is most convenient. Although there is a YouTube link also, and there could be some questions coming there. So I thought maybe I can review some of the points related to the general course and also um, try to answer some of the questions. And if any of the questions remain unanswered, then of course, we do have the discussion forum still, so we'll try to address those. So I think maybe you have already met the TAs, but let me just introduce. So I am myself Professor Shikha Laluria from the Department of Biochemistry. And the TAs this year are Swagatinath Gopinath and Anshul Kumar Jaryal. And it's Anshul's first year here for the course. So we are quite enthusiastic about interacting with all of you. And I think also this year we have got some PMRF fellows. So I believe you would have been interacting with them. They have been holding the separate, live sessions or tutorials for the course. So that's good. I mean, I hope everybody's up to speed. I think not noticed much problems in the assignments. People have been doing well. And of course, many of you I know are enrolled for the purpose of learning. So that's always good. So I think I have a small PPT which will remind me what are the points that I just wanted to tell all of you about. And then I'll address a couple of questions. Before that, I would like Anshul and Swagat to sort of turn on their videos and say hello.
Visible.
Yeah, please introduce yourselves. Swagat, your audio is off, I think. Okay, Anshul, you can go ahead.
Yeah. Hi, am I audible now?
Yes, go ahead, please, quickly. Then we'll go on with the session.
Oh, yeah. Yeah. So I'm sorry, I muted myself. So this is Swagat Nath, and I'm one of the TAs for this cell biology course taken by Professor Shikha Laloria in the NPTL. And welcome to this course. And I mean, you guys are going to give exam still. So uh, hope you guys have had a happy learning and hope that it inculcates some good points in your life and it improves your some educational prospects, hopefully. Uh, Thanks and happy learning.
So, hello everyone. My name is Anshul Kumar. I have joined this year only as a TA for this cell biology course. So, I hope this course will help you understand about the cell biology in detail, and wish you guys best of luck for your exam.
Okay, thank you guys. I'm going to share my screen also. That we have something to talk about. Okay, is this visible to everybody?
Yes, ma'am. Is it's visible to us?
All right. Yeah. So these things we have covered, you all are here already. So there are some points in the discussion forum, some things are not clear to everyone, but there are the links to the tutorial by the PMRF tutors. So that is on the lower left-hand side, and it's called problem solving session. And also there are clarifications to your queries. And many announcements related to the content will be there. And of course, some of you have been participating in the Ask a Question section. Other thing I want to say is there are text transcripts already there on the platform, so you could look at them. But in addition, some of them may not be in proper order. So I have taken the trouble of posting the text transcripts weekly on a weekly basis. So I think by now someone had asked for that for the last one, but I have posted all of them week-wise in the discussion forum Q&A part. So please look for that message and the attachment. Okay, so that will help you understand, in case we have a weird accent or something. Okay, so this is the link where the PMRF tutors are Isaac Cherian, Shrishti Mandal, and Sonam Pahuja. And, do interact with them and you can ask your doubts. I think they've probably covered only the last week is left. Okay. Um, you may have some queries. I think in the portal itself, you can get this information. Some of you have registered for the exam. So you will get a certificate if you pass the course, and the best 6 out of 8 assignments will be considered. And of course, any of the details you can just check it out at the portal itself. And I too wish you very good luck for the upcoming final exam. So, yeah, I have made a list actually of the yeah, I made a list of some of the questions that were coming up just to start to warm up, and I think TAs also have something to say at this time. So let me see if I can access the Word file here. So there were some questions. I think some questions about ATP. Soumita Das had asked a question. So she is curious whether, in which stage of the cell cycle is ATP synthesized? And we did answer it to some extent in the forum, but I'll like to elaborate on it because I do think it's an interesting question. And actually, this is something interesting. The cell needs ATP constantly. And I think there is some basal level of ATP synthesis going on throughout the cell cycle, as Anshul has also answered to you in the discussion forum. But nevertheless, we looked it up. And the different phases of the cell cycle, G1S, G2, and M. So the gap phases, G1 and G2, are the phases where the cell is basically preparing for the next active phase. In one case, after G1 is S phase, and after G2 is mitosis, which is a very energy requiring process. So there is basal ATP synthesis throughout, and there is ATP in the cell. But as per many of the reports, I came to conclude that in G2 phase, there is more ATP synthesis. And here's an interesting reference. It's actually a theoretical analysis, but you should look it up if you're interested. In this, they've actually analyzed, how is metabolism coupled to the cell cycle. So for example, in this graph, you will see the ratio of ATP to ADP here, how it fluctuates. I don't know if you can see my pointer. Okay. So you can see in mitosis G0, G1, S, and G2. And basically, in this arrow, up arrow indicates that this ratio is increased. So both in S phase and in G2, they are indicating there's more ATP around than ADP. Okay? And of course, in mitosis, there's a lot of ATP hydrolysis happening. So that's why the ratio falls down. And I'm sure there's ongoing ATP synthesis all the time. So I hope that clarifies. I've given the reference, so you can look it up. It's much more detailed than what I said. The next point interesting I found in this paper, or rather it's kind of like a theoretical analysis. So they have proposed that maybe there is variation in intracellular pH, and that may affect the metabolic activity, the activity of the different enzymes. So here you can see in G2, they have indicated that ATP synthetase and fatty acid synthetase would be, or they are expected to be more active in this phase. Okay. So I hope that answers your question. And Soumita Das, okay, and other students also who are in the session. Now, there was another question which is also related to ATP synthesis. It's a fascinating process, of course. We discussed in detail in the course about how ATP is synthesized and the structure and mechanism of the ATP synthase, which is this fascinating molecular machine in cells. So, after a lot of thinking, and one of the students has asked me, this is, let's see, this is Reema Deep. Reema Deep, I don't know if you are there. So the question here is that in bacteria, which is a prokaryote, they're saying, they're asking that mitochondria are not there. So how will you get the energy? How would the ATP be synthesized in this case? So of we also talked about the endosymbiotic hypothesis of origin of mitochondria, that it's thought that they are in fact derived from bacteria. The mitochondrial inner membrane has got the ATP synthase. So in case of bacteria, of course, they also have ATP synthase, and that is in their membrane. So in that case, it is oriented such that the F1 part of the molecule, it is facing the cytosol side. Okay. So in that case, the ATP would be synthesized and available in the cytosol. It will be released into the cytosol. And I'll quickly show you one diagram that I came across, which is showing this point very nicely. Okay, I'll get into slideshow view. So here you can see this purple or lavender colored molecule here. That is the ATP synthase. Yeah. Okay. So this is a membrane here, and this is the cytosol part. You can see my pointer. So this is the ATP synthase of the bacteria. It is in the membrane of the bacteria, and so are many other. The electron transport chain components are here, and the gradient is generated across this membrane. Okay, so it's nothing very puzzling actually. It was already there in bacteria, and we can see that the eukaryotic cells have inherited it, or it's and their ATP synthase is evolved from the bacterial precursors or ancestors. Okay, so yeah, this is beautiful. It's from an article by Boyd et al. And do look it up. But otherwise, the mechanism is similar, and there's a lot of analysis about the mechanism, both at a structural as well as a functional level available. There's a huge amount of literature, so I won't be going into that. All right. So coming back on time. Right. I had noted down some other things. I think I'll stop sharing now. We can see what you guys are curious about. Okay. Yeah. So let's see what else is there. I just mentioned this. Yeah. There was a question about RBC. Okay, that's by Rama Devi. So let's see who all are there. Any of the people who ask questions are there. Yes. So anyway, Rama Devi's question was in the very beginning. We've not gone through all of them, but whatever I could manage so far, others I will answer online. So she has asked us that RBC, how do they survive without DNA? How do they work without a nucleus? That's the question. RBC, as you know, they are enucleated. They don't have a nucleus. So, of course, it's a valid concern. How do they survive? How do they work without DNA? And I did give an answer in the forum about this, a very general one actually, that you all might be aware of also. And that is that the RBCs, their nucleus, it was there in the original cells, in the progenitor cells, but during the development, the nucleus has been lost. And there's a particular mechanism of that, quite fascinating. But the question is, how do they survive without a nucleus? So the thing is that RBCs are not there forever. They're not long-lived cells. They are relatively short-lived cells. They have a limited lifespan. So yes, they will not survive forever. But their main function really is to transport oxygen, and they want to be very efficient in that. So, people say, yeah, you can think of them as sacks of hemoglobin. So when you lose the nucleus and some of the other organelles, there's more space for hemoglobin, which is the iron-containing molecule that binds oxygen and helps in its transport. And then by losing all this bulk inside, the cell also has a distinct shape, a biconcave flat shape. So this helps in, for it to move around more, it's also a little more flexible. So it's got a high surface area to volume, and this helps in the gaseous diffusion that it needs to do. And these cells, they also need to squeeze through the narrow blood vessels and capillaries. So being thin and flexible, less bulky helps in that indirectly. So if the cell had to survive very long, yeah, we would need DNA because at some point it will run out of the macromolecules and there's degradation and all that. They do have a limited lifespan and new ones are continuously being made. Okay, so having said that, try to look up just to satisfy the curiosity, how does this process happen? And indeed, some labs have studied this process. I came across a paper from the Lodish lab where they looked at a model system, cultured mouse fetal erythroblasts. And there they studied this process. And they found that they were culturing them, and then after the fifth division, they could see the enucleation happening. And the more interesting part I thought also was that the cell which remains, like, so there is a division where you get one enucleate cell and the other cell gets the nucleus. So it has got twice the amount of DNA, it gets all the DNA. But that cell, it gets eaten up by macrophages. That was something interesting mentioned in their work. So, it's something quite fascinating process. Now, while looking into all of this, I came across that RBCs, actually nowadays people have found very interesting new things about RBCs that were not known earlier. And people think maybe they have some other functions also, just beyond the canonical, well-described function of oxygen transport. So some researchers in the last couple of years really have found that indeed inside the RBCs there are some DNA fragments, like there are some long DNA fragments, and when they analyzed them, they found that they were from other cells. So RBCs could actually acquire DNA from other cells, including tumor cells, and also from some pathogens such as bacteria and so on. So this is a very new and active area of study. And it's fascinating, but this is not the original DNA which was there in the RBC progenitor cell from which it is derived. Okay, so that's what I have to say pretty much. And I think the TAs may like to say something you all have written. Let me see. Yeah, at least you all can say hi to us and overcome your hesitation. Are you all well prepared for the exam? Somebody was asking me in the forum what kind of questions will come. And I think we made it pretty clear the questions will be mostly MCQs and MSQs, that is multiple choice questions, which may have one or multiple answers. Because given the large number of students in this course, it's not really possible for us to assess the descriptive type questions. And I think you all have been doing well. So please study whatever was taught in the class. And if you do that and understand all of that, you stand a pretty good chance to do well. So there are some new students. Prachurya Das, welcome. And somebody else. There's a query that you were not able to submit the first two assignments. But as I mentioned, only the best 6 out of 8 will be taken. So I hope that you would be able to get be considered, yes. But I cannot comment on individual person's marks here. Itishri. I think Pratik also had a question he has left, so we will address it later. So we do have some time. I think I can ask maybe Anshul to talk a little, if recap some of the content. That may be a good thing.
Just give me a second. I'm about to share my screen. Then I'll present a small presentation. We'll try to recap the key highlights of this course, and hopefully, this recapitulation will be helpful to all of you.
Are there any other queries or questions? So you can definitely write in the forum. Okay. So I hope students are listening, and I think maybe this year there's been a lot of help available, so there are not many questions. Is that correct? Say something. Okay.
Yes, ma'am. We are listening.
So, ma'am, shall I start the presentation?
I think there's also a message from NPTEL that if there are no questions, we can wind up. So that option is also there.
Okay.
I just wanted to hear from the students, if they have any doubts or something, we can try to. I mean, certainly we are still tuned to the discussion forum, and I know there are one or two questions left, so I'll be addressing those over there. But here, I think it's nice that you all have joined. No one has any particular worries about the course. I think everybody is up to speed and doing quite well. So we are happy to know that. I may share some references the forum, and you can actually look them up, but it's not compulsory. Like, don't think that they'll be a bit advanced, so won't be coming in the exam, but it's good to look up, and including the two references I spoke about today. So Anshul, do you want to go ahead, or any of the students want to say anything? I think there are one or two unresolved questions in the forum. I'll address them there. That will be a bit detailed. And other questions were just logistical questions, like I hope the NPTEL staff are seeing this, that somebody could not submit two assignments. But I think since we're only taking 6, they'll be able to do that. But we can look into that. Um, Maybe we can wind up. Anshul, do you want to go ahead with your presentation?
Ma'am, I think we should ask the chat if their students are interested. Then we can go ahead. Otherwise, it's up to them. We can wind up if they
Yeah.
want.
Students, please write something. Your TA has prepared a small recap, like I guess you're doing that with PMRFs also. So, do you want us to do that? What type of questions will be there in the final exam? Anup, all MCQ. Yeah, they'll all be objective type. There'll be MCQ, and there'll be another category where there may be multiple correct answers. So there you have to select the correct answers, and if you select a wrong one, then there is some negative marking also. So those two. Yes. Anybody else? I think Anshul, you present couple of slides. I don't know what you have, maybe some of the key points you have mentioned. So I'll give you some 5 minutes and then we'll see again. Okay.
Yeah, sure, ma'am. Just give me a second. We'll again share. I'll again share my screen.
Let me check this. Payal was asking about the previous lecture PDF. So I have actually already shared all of them. Okay. Let's see what Anshul has up his sleeve. Final exam, wrong answer have negative mark. That's what Itishri is asking. No, I don't think that is correct. In general, it's not, I don't think there's negative marking. You please see whatever instructions are there in the platform. But there is a category of questions where there is MSQ, right? So there'll be two correct answers, or there may even be three correct answers, and then there are marks for each of them. So actually, I'm not really sure whether there's a negative mark, but you may, if you select, one correct and one wrong answer, you will only get half. Okay, so I don't think there's any need to worry too much. Just try your best to give the right answer. And think a little, like sometimes it's a little tricky. We'll have some questions similar to assignment question, but maybe a bit more complex questions also. Over to you, Anshul.
Yes, ma'am.
Yeah, just go to the main things and maybe take 5 minutes only. And meantime, students can think what else they want to ask. I think somebody, Pratik, had joined and left and came back again. And what I wanted to say, there are some pending questions in the forum, so I'll answer them right there.
Yeah, so we have prepared this small presentation. It will highlight some key topics from this course, and this will help you brushing up your memory, and hopefully it would be helpful for you because your exams are like coming soon. So I'll try to give a
Anshul, you can turn on your video. I'll switch off mine meantime. Hmm?
okay.
And try to finish in 5 minutes.
Yeah, sure, ma'am. One second.
Yeah, meantime, people can write more comments or anything in the chat box. You turn on your video and there's not enough light, so maybe you need to reorient yourself.
Am I visible? Yeah, so I'll proceed now. So we'll give a brief overview about the organization and the component of the cells, DNA as a genetic material, the mechanism of the DNA replication, regulation of the cell cycle, meiosis, apoptosis, cell death, cancer, and plant cells and stem cells. So we'll start with the organization and the component of the cells. As we all know, cells are the fundamental unit of life, and they have an organized structure composed of various components that perform specific functions. So broadly, we can classify cells into two categories: the prokaryotes and the eukaryotes. The eukaryotes mostly like all the multicellular organisms and like plants and animals, and prokaryotes, as we know, that consists of bacteria and archaea. So, regarding the organization of the cell, cells can be organized into basically into three main parts. The first one is the cell membrane, which we also call the plasma membrane. It's a phospholipid bilayer with embedded proteins that surrounds the cell. And then it also helps in regulate the transport of the materials in and out of the cells, and it maintains the homeostasis and enables the cells to communicate. Also the then this membrane then comes the cytoplasm, cytoplasm. It's a gel-like substance within the membrane that contains the organelles and the molecule. And then finally, we have a nucleus, which is the controlling center of the cell and contains the DNA. So uh, we'll discuss about the membrane bound organelles. These membrane bound organelles are specific to the eukaryotic cells only. For example, nucleus that I have discussed in the previous slide. Then we have mitochondria, which we all know is the powerhouse of the cell. It generates ATP through the cellular respiration. Then we have endoplasmic reticulums of the two type, the rough endoplasmic and the smooth endoplasmic reticulum. The rough has ribosomes and it is involved in protein synthesis, while the smooth endoplasmic reticulum, it lacks ribosome, but it is involved in lipid synthesis and certain detoxifications. Then we have the Golgi apparatus. It is involved in modification, and it sort of and it sorts the protein and packages protein and lipids for transport. We have peroxisomes, which are involved in breakdown of the cellular debris, and then there is another important organelle, chloroplast, which I have not shown in this particular diagram, but it's specific to the plant cells. It helps them to carry out photosynthesis to convert sunlight into the energy. Then, of course, we have non-membrane bound organelles. These are present in both prokaryotes and eukaryotes, like ribosomes in the cytoskeleton. Ribosome specifically is involved in the proteins synthesis, while cytoskeleton, they provide structural support and they allow cell to like it enables cell for movements. And then also we have external structures like cell wall, which is present in plant, fungi, and bacteria. It provides the structural support and protection. And we also have flagella and cilia, which aids in movement. A typical example is like sperm cells, which have flagella. And then we also have the prokaryotic cells like bacteria and archaea, which lacks nucleus completely. So they don't have membrane bound organelles, but they have ribosomes, plasma membrane, and the cell wall. So, DNA. Next, I'll discuss about the DNA as a genetic material. So, DNA is deoxyribonucleic acid. It's a fundamental molecule responsible for storing and transmitting genetic information in all living organism. It plays a crucial role in heredity, encoding instructions necessary for development, functioning, and reproduction of the cells. So the DNA is a double helical molecule composed of two strands bound around each other, and it consists of nucleotides. Nucleotides are the basic unit of the DNA, each containing phosphate group, deoxyribose sugar, and the nitrogenous base like adenine, thymine, guanine, and cytosine. So, and this adenine base pairs with the thymine, cytosine base pair with the guanine through hydrogen bonding, and this is what we call as the base pairing rule. So, and also one important thing, this double helix model, it was proposed by the Watson and Crick in 1953. So regarding the functions of the DNA, one of the main function of the DNA is the replication. So DNA replicates before cell division, and it ensures the genetic continuity. And then another important function of cell of DNA is gene expression. So basically, this DNA, one second, yeah. So DNA is first transcribed into a single-stranded RNA, which then is translated into proteins, and these proteins are the one which determines the cellular function. And also, another key aspect of DNA is the genetic variations that it undergoes. The mutation in the DNA sequences contributes to the evolution and the diversity among species. And I'll quickly go through the mechanism of the DNA replication. So it's yeah. So it occurs in three phases. The first one is the initiation, and then yeah, so it occurs in three phases: the initiation, and then elongation, and the termination. So during initiation, the helicase will unbind the double-stranded molecule, and this replication fork forms, and where the DNA polymerase will act upon and synthesize this the leading strand, which is the continuous strand, and then on the other strand, which we call it call as the lagging strand. Uh, uh, Sorry, so DNA, basically what happens is the DNA polymerase synthesizes the new complementary strand by adding nucleotides according to the base pair rules that I have mentioned in the previous slides. On the leading strand, the replication is continuous, as can be seen in the diagram, and while on the lagging strand, the short DNA fragments are added. The short fragments are called the Okazaki fragments, are synthesized and later joined by the DNA ligase. And finally, in the termination, once this replication is completed, termination occurs. So this ensures that the genetic information is faithfully transmitted to the daughter cell with minimal errors. Okay, so another important thing that we have learned in this uh, course is the regulation of the cell cycle. I won't go into the detail of this. So I hope I'll quickly uh, bind up this slide here only, and that's it. Yeah. So I hope you guys have it helped. These slides have helped you guys to brush up your memories.
Thank you, Anshul. So is there any other query, anything else that we can help you with? Yeah, everybody's curious about the exam only. How many questions are MCQ? How many MSQ? Frankly, I don't remember, but majority will be MCQ. Others would be MSQ. It really doesn't matter. You don't need to know that in advance because you have to look at the choices provided. But it will be indicated in the question paper. And I think this is the last week, the 8th week. So do well in the assignment and stay tuned to the discussion forum and even the announcements because we may share some more references and so on and answer some of the remaining questions also. So were most of you attending the tutorials? We had weekly tutorials this time. Okay, I think there's no further questions. So I think we can wind up now. And please stay tuned to the discussion forum. And if you have any additional doubt that you forgot to ask today, please feel free to put it there and we'll try our best to answer it. Okay. We'll close this live session now. Okay, thank you. NPTEL team is also with us, so they will help in the technical aspects. Please close the session. Thank you, everyone. Good luck on your exams.